Hello everyone, it's Lauren Bean. We're going to go over chapter 19, which is the blood vessels. And as you can see from this image here, the blood vessels are an elaborate network of tubes that are transporting blood to and from the heart and to the tissue cells to provide nourishment and to remove any waste that accumulates at our tissues as well. You want to be considering our pulmonary circuit and our systemic circuit that we talked about in the heart chapter. Vessels are what are responsible for creating the actual circuit, right? Allowing for the distribution of blood to the lungs or to the rest of the body. Okay, we're going to be starting by looking at blood vessel structure and then get into more the physiology of how it all works. So for the blood vessels, they are dynamic tubes that transport blood to and from the heart and the body tissues. So this is going to include arteries, veins, and capillaries. Those are the three major types that are involved. And as you can see with this image here on the right-hand side, we have the heart being shown. So here's our pump. Arteries are what take blood away from the heart, right, to nourish the body tissues with oxygen, nutrients, hormones, etc. Capillaries serve as the site of exchange where that true transfer is taking place. Capillaries are very, very small, allow for things to actually move in and out of the blood vessels. And the veins are what return that oxygen poor blood or deoxygenated blood, sometimes that's said, back to the heart. Okay, we're going to look at the different types. You can even see labeled here in this image, elastic arteries, muscular, so on and so forth. So there's different types, but you want to first think about the general idea. What's also important down below here I have listed and is shown in green here is the lymphatic system. This is responsible for collecting fluid that is what we say lost at the capillaries. So during that exchange, that shift in fluid going out to nourish and the return of fluid coming back in containing the waste, sometimes or all the time, there's a little bit of fluid that is not collected on the venule side. And the lymphatic system is responsible for collecting that fluid, transporting it through the lymphatic system, monitoring the fluid as well for any pathogens, cancer cells, etc., and then returns that to the general blood circulation. So we do have the lymphatic system following along with our blood vessels, and we're going to talk more about that in the next unit when we talk about the immune system as well. So for blood vessel structure, the innermost layer is the tunica intima, and it's intimate with the blood. It forms the lumen of the blood vessel. So the lumen is the hollow cavity where the blood is actually flowing. So we'll use the term lumen quite a bit in this semester, anytime you're talking about A&P, and just think of it as the inside of a tube where stuff is flowing. So it can consist of multiple tissues. It's all going to kind of vary depending on the size of the blood vessel we're looking at and where it's found. There's always going to be an endothelial layer made of simple squamous cells forming that whole layer. And then there'll be a subendothelial layer, like a basement membrane, just providing some connective tissue support. And it can have an elastic membrane around that as well, allowing for some stretch. The tunica media is composed of smooth muscle cells and elastic fibers. So this is the middle layer, it's the muscular layer, right? And that can also have a supporting elastic membrane as well. The tunica media is really what's responsible for providing the bulk of an artery, and the wall of an artery is typically much thicker than what we see with a vein. So you can quickly kind of see that, how the tunica media is thicker here in the artery and thinner over here with the vein. The external or tunica externa layer mostly being collagen fibers, just providing structural support. And when we have a really large vessel, there'll actually be a small network of blood vessels supplying that tunic as well. And we call that the vasa vasorum. So blood vessels can be really, really big and need their own supply of nutrients and exchange of waste. So we may or may not see that vasa vasorum present there, depending on the size. Okay, and then with the vein, we're seeing similar layers, similar tunics, but a little bit of variability there. It's going to lack an elastic membrane. Veins are lower pressure. The blood is not surging through the veins like it is in an artery. It doesn't really need that extra elasticity present in the tunics, and so we don't see that really being found. Again, the tunica media is thinner. The wall of a vein is thinner than what we find in arteries, but it will have a very thick collagen tunica externa layer, providing a lot of structural support. In our medium-sized veins, we also see that the tunica intima turns inward to produce valves. So valves are unique to veins, right? That is important to return blood back to the heart. Veins, again, are low-pressure vessels. The pressure of the blood inside is not very great, and it needs to work uphill most of the time to return to the heart. Right? You think of veins like in our leg. Well, all that blood has to work against gravity to get up to the heart in their chest. And then down below here being just a capillary, my picture is kind of cutting it off, but the pink being the endothelial layer, which is then supported by a basement membrane. So here we have a whole network of capillaries form a capillary bed, but each tiny little tube is a capillary. And again, this is a site of exchange where we find nutrients and waste, oxygen and carbon dioxide moving between the blood and the tissue fluids. So for a tunic, that's just the layer or the wall in the blood vessel, and those are surrounding the internal space called the lumen. So the lumen is just, again, the cavity inside. We could be talking about, the GI tract, that has a lumen, blood vessel has a lumen. It's just the hollow inside part of the tube. Here I've listed for you just kind of in words what I just talked about on the previous page. I just always want to make sure that I have a visual, but I have all this information written down for you. I want to elaborate a little more on the tunica media here. That smooth muscle layer is innervated by the sympathetic nervous system. Only the sympathetic nervous system, okay? There's no neurons from the parasympathetic here. Only sympathetic vasomotor nerves innervate this smooth muscle layer, and they're responsible for causing vasoconstriction and vasodilation within the tunica media of arteries. So if we constrict a blood vessel, that's going to decrease the lumen. It's like we're squeezing to a tube. You take a hose and water going through. If you step on it, you kink that tube, right? You're constricting it, causing vasoconstriction, decreasing the lumen diameter, and of course that will disrupt flow, right? And then vasodilation is when the vessel dilates, the lumen gets larger, right? And more blood can flow through that vessel. So the sympathetic nervous system has a great control over blood flow, blood pressure, due to the vasoconstriction and vasodilation that takes place in the tunica media. So we're going to look at the arterial system, focus on the different types of arteries that are present, and it's going to be clear why they're named what they are. It all makes sense when you consider the function. So for the largest arteries, we call them elastic or conducting arteries. And this is going to include the aorta. So you see the aorta here coming off of the heart and its major branches. So the brachiocephalic trunk, left common carotid, a left subclavian artery. So those are very large arteries. There's a lot of elastic present. And that should make sense to you because as blood surges out of the left ventricle and enters into the aorta, there's a lot of pressure there, right? It's being ejected. It's surging out. So those vessels are going to stretch to accommodate for that incoming blood, then recoil and move the blood forward, okay? When we look at the tissue, when we look at the tunica media, these blackish blue little squigglies are representing elastic fibers, allowing for the stretch and the recoil. Stretch, recoil, stretch, recoil. That's what actually creates kind of that pulsatile flow of blood, is that elastic tissue there. And they're conducting the blood away from the heart, okay? These are pressure reservoirs, right? And that correlates with, the stretch and the recoil. Stretch, recoil. conducting, surging the blood, moving it forward due to that assembly of the elastic fibers, keeping the flow of blood continuous but also pulsatile. These large elastic conducting arteries are not involved with vasoconstriction. That should also make sense. There should be no reason that the vessels are constricting in that area. It would actually be very dangerous because it would create a lot of afterload, create a lot of back pressure on the heart. That would not be good at all. So no, constriction, dilation, sympathetic nervous system control really occurring in these large arteries. Then the muscular distributing arteries, these are distributing the blood to major organs, major tissues, right? So these are going to be the middle-sized ones. A lot of smooth muscle being found in that layer, right? So are going to be active in vasoconstriction, not really seeing a whole lot of elastic fibers here. So, not being involved with the stretch and recoil, it's more like, okay, now let's pump the blood where it needs to go, right? But muscular because it's abundant in smooth muscle. And this accounts for a lot of the named arteries that we think of, the femoral artery, the renal artery, so on and so forth. The ones that we give names to or we label on a diagram, those are majority going to be muscular distributing arteries. Then we have the arterioles, also called resistance arteries, because this is where the blood meets a lot of resistance. We have quite a reduction in size taking place when we go from these elastic conducting down to the muscular distributing, and then these tiny arterioles. The blood is meeting a smaller lumen, and with that comes an increase in resistance, right? It's not going to allow the blood to flow as easily because it's getting smaller. Right? It's like taking a garden hose and having water in it, flowing the water through the big garden hose, and then making the water flow through a tiny little straw, right? The water would meet a lot of resistance doing that, and that's kind of what happens at that transition of blood to these smaller vessels, okay? Also, within the arterioles, the smooth muscle layer is very active in vasoconstriction and vasodilation. So really going to be controlling and dictating where the blood goes. Is it going to go all throughout that blood capillary? Is it not? So on and so forth. So this is the primary site of vascular resistance because we have that reduction in size, increase in resistance, reduction in blood flow. And this becomes very important because we don't have enough blood in our body to nourish all of our capillary beds. At any one time, 3/4 of our capillaries are shut down and are not active, and blood is not just flowing through all of them. Our body, our brain can decide where the blood needs to go at any given time, depending on what we're doing. Right? Are we running? Are we sitting on the couch? We just ate a big meal? Are we sleeping? Whatever, right? Blood is shunted to where it needs to be for that particular moment in time. Capillaries, so smallest blood vessels, really, really, really tiny. So tiny that only one red blood cell can pass through at any given time. Red blood cells about 8 micrometers across, so that's about the diameter of the lumen of a capillary. Just endothelium and a basement membrane, so lacking the other tunics that we see in the larger arteries and veins. This is very important because it allows for things to be exchanged, right? These are exchange vessels. Exchange could not occur if you have a big layer of smooth muscle and a big layer of collagen, right? That does not allow for diffusion. These endothelial cells are very good at diffusion because they're simple squamous cells. They are flat, squashed, fried eggs, right? Very thin, very efficient for diffusion, right? There are some other structural components that contribute to it. The cells are held together by tight junctions. They make intercellular clefts. I'll have a better visual of that coming up. And what we're going to talk about now are the 3 types of capillaries found throughout the body. So first, here being continuous capillaries, we look at the structure. Here are endothelial cells. They kind of overlap one another to create that layer, to create that tube. They'll be held together by tight junctions, so that stitching of the quilt anchoring the cells together. Areas, there's going to be a little bit of a cleft, a little bit of a gap or intercellular cleft that allows for larger particles to pass between the capillary wall. And then they can be supported by these pericytes, cells that kind of wrap themselves around the capillary and just, serving as support. Can serve as a stem cell to help repair, can help to control permeability, help control, the healing of a broken blood vessel as well. Tight junctions are really involved with creating a tight barrier. So these continuous capillaries are the least permeable. Okay, they're held together very tightly, so only small particles are going to be able to pass through these types of capillaries. They're not leaky, like we say, but these are the most common. So widely distributed throughout the body, right? Ensuring that, the important stuff gets in and out, but not allowing, big proteins and other stuff to pass through the capillary wall. In contrast, here with the fenestrated capillaries, there are fenestrations. There are pores that are pretty good size and allow, larger particles to pass through the capillary wall. So these have an increased permeability, or we say these are a little bit more leaky than the continuous capillaries. These are going to allow for rapid diffusion, right? More stuff is going to be able to filter through this capillary wall. So these are very abundant in the kidneys. So the kidneys are responsible for filtering our blood, right? And in doing that, their structure contains these fenestrated capillaries so that filtration can be adequate. Also going to be abundant in the intestines, where a lot of absorption of nutrients is taking place. And then in endocrine glands, right, producing hormones, well, that needs to be able to get into the bloodstream. So the fenestrated capillaries are going to allow for that to occur a little easier. Then the third being the sinusoidal capillaries. These are the leakiest of all. These are highly modified. There's huge intercellular clefts between the cells. There's these huge holes even in the endothelial cells themselves. So very porous. You can see there's an incomplete basement membrane as well. So very large particles being able to move between these capillaries, right, in and out of the bloodstream. So these are isolated to unique parts of the body. The classic example would be the red bone marrow, right? That's responsible for producing all of our blood cells. Well, as the blood cells are being synthesized in the marrow, it needs to get out into the bloodstream, right? So it needs to be able to exit the bone marrow, and these sinusoidal capillaries allow for that to take place. Also abundant in the liver, adrenal medulla, the spleen, where we're going to see, similar things taking place, where we need to have efficient, diffusion occurring. The spleen stores a lot of extra red blood cells and white blood cells, so there's movement to and from. The liver is moving large proteins, different nutrient components to and from. So it kind of makes sense why we find those in those unique organs in the body. Okay, so continuous are widely distributed throughout, and then the fenestrated and the sinusoidal capillaries are isolated to unique organs and tissues carrying out unique functions. So now we'll talk a little bit more about capillary beds, get a little bit more technical with the terminology here. So when arteriole becomes actually a terminal arteriole, so the arteries are terminating at this point. So a terminal arteriole feeds into a capillary bed. And that can contain 10 to up to 100 capillaries within that bed or within that network. Purple is telling us we have an exchange of oxygen and carbon dioxide taking place. Blue now saying, okay, this blood is deoxygenated. And we have a post-capillary venule collecting that, then a venule, then a vein, vena cava, back to the heart. But it's post-capillary, right? It's at the end of the capillary bed. It's the post, not the pre. So you could think of the pre being the terminal arteriole, blood flowing through that first, then to the vein or venous system. Okay. So again, there's a lot of variability in the size of the capillary bed, how many capillaries are contributing to it. But like I just said earlier, at any given time, about 3/4 of our body's capillaries are shut down. We do not have enough blood in our vascular system to fill every single capillary bed. And it doesn't even make sense to do that. It would be a waste of blood, of time, of energy, of nutrients, right? Our body can control where the blood is going, what capillary beds are being supplied, depending on the body's needs, right? When I eat a meal directly after that digestion is taking place, I'm going to have a lot of blood flow go into my gut to do that, right? But, if I haven't ate in 3-4 hours, I don't need a whole lot of blood flow going to my GI tract, right? So, the flow of blood, where it is controlled depending on what needs to happen at that time. Local chemicals are able to control that. Our sympathetic nervous system will be able to control that, right? The sympathetic neurons are what innervate the smooth muscle layer of our vessels, control vasoconstriction and vasodilation, closing off a capillary bed or opening it up. So here you see one that would be like open, blood flowing through. Here's one where the terminal arteriole is constricted and the blood is just bypassing that capillary. It's just not needed at that time. It's just going to be shunted and bypass it. And then, at a later date, it may open up and blood flow through it. Right? So there's a lot of change. There's a lot of things that are going to really determine that. So to be able to understand that idea of bypassing capillary beds, we need to talk about anastomoses. So in general, vascular anastomoses, which kind of includes all types, is when there's some continuous connection between two blood vessels. And capillaries are not really being involved with an anastomose. And there's different types. So arteriovenous anastomose, connecting an artery to a vein. And this is like I was talking earlier, that idea of, the blood being shunted, bypassing that true capillary network and, just going right through artery to vein and kind of skipping that whole capillary exchange. Or we can have arterial anastomoses, so just two arteries kind of being connected, providing an alternative pathway, providing like a detour. This becomes important if, an artery is blocked or occluded in some location. Well, if the blood has an anastomose that I can take, a detour that I can take, that can help to make sure that the blood is still being nourished with what it needs. So we find these like in our joints, abdominal organs, brain, and the heart, areas where an occlusion could be really detrimental. So there's a detour there. Then for venous anastomoses, these are extremely abundant. There's many, many detours that blood can take within the venous system on its way back to the heart. So much so that a blocked vein rarely ever disrupts blood flow, rarely ever really causes a problem. If someone has varicose veins, which I'll talk about shortly, we can just take that vein right out, just remove it, and it's not a big deal because there's just so many detours that blood can take within the venous system. Okay, so now we'll talk more about that arteriovenous anastomose in relation to the capillary bed. Okay, so when I was talking earlier, I was saying how, we can kind of shut down this capillary network and prevent blood flowing through the whole bed. And this just is showing us in more detail what actually is really taking place there. Okay, so arteriovenous anastomose is connecting an artery and a vein, or more specifically an arteriole and a venule. Okay, and there is a vascular shunt involved in that transfer, or that's the true anastomose here. With the vascular shunt, we have a meta-arteriole and a thoroughfare channel, right? And that is the shunt, that is the bypass. So instead of blood flowing through this entire capillary network out here, it just surges right through the center and it shunts its way through. What controls the blood flow either through the shunt or through the entire capillary bed are these precapillary sphincters. Just really a cluster of smooth muscle cells found right along the vessel. And if they are stimulated to contract, they're going to squeeze and constrict the capillary and prevent the blood from flowing out into the network. Or at other times, they're dilated and blood can flow through that network. So really, it's the precapillary sphincters that are regulating that flow. In the next, segment of this chapter, we'll talk about what really controls that, the chemical conditions or the sympathetic nervous system, how it really regulates that blood flow. Now for the venous system, so veins, right, return blood to the heart. We typically see them as blue, and that tells us that they are oxygen poor or deoxygenated blood. It's really not appropriate to say deoxygenated because there's still oxygen within the blood in the venous system. It's just less than what we find in the arteries, right? But we do have kind of a reserve of oxygen within the blood at all times. Okay, it's not like we zap all of it out in the capillaries. There's still oxygen remaining. But since the hemoglobin doesn't have as much oxygen bound, there is a change in the color of our blood that literally does take place in the body. So we emphasize that with red and blue. So in general, veins have thinner walls, they have thinner tunics, and they have larger lumens. Veins don't really retain their shape very well. Like when you look at an artery, it looks like a nice tube, the lumen really retains its shape. It's nice and circular. With a vein, it tends to be kind of collapsed. And that's just due to the fact that they are thinner walled, large lumen. They just don't really retain their shape as well. And a lot of that is due to the fact that the blood pressure within veins is much lower than what we find in arteries. That flow and that pressure has dissipated as the blood has gone through the arteries, gone through the capillaries. Now, going through the veins, working uphill to get to the heart most of the time. There's just not a lot of pressure inside of these vessels. However, they do have the ability to store a lot of the blood volume. So they are capacitant vessels. They hold around 65% of our blood volume. Okay. So they're kind of, like storage vessels. The blood is still moving and going through, but, the lumen's larger. They have the ability to kind of retain that volume at a much larger scale than in the arteries. So we have large and medium veins, and then our venules. There's different terms we could use to describe that, but in general, large, medium, and then there's venules. For the large veins, you're going to include like the vena cava, the pulmonary veins, internal jugular vein, which runs along our neck, and then the renal vein, collecting blood from the kidneys. Very big, large lumen, 10 millimeters up to 1 centimeter, which is a decent size there. Medium veins are smaller, and what's unique here is that they have venous valves. So they have inward projections of the tunica intima or interna, and that creates a valve. Very similar to what we saw with semilunar valves, right? Kind of the cup-like arrangement. So as blood, it surges up, but then it kind of falls back, and those cups collect any blood that kind of drops backward and ensures unidirectional blood flow. And then for the venules, some are, muscular venules, so they have a little bit of smooth muscle present there. And then the post-capillary venules, those are the really, really small ones that are collecting blood from the capillary network. These also tend to be quite porous. So they're very good at collecting fluid, collecting proteins and any cells that may be in the tissue, right? So they're on the receiving end of a capillary bed. So their structure is different than what we see in, say, like a terminal arteriole, which is just releasing the fluid. So veins, again, they do have valves, and it's very important for unidirectional blood flow back to the heart. So there's not a lot of pressure here, right, pushing the blood forward. So there are other things that do help with that return. The squeezing action of our muscles as they contract and relax can push on the veins and encourage the blood to move forward, right? And while the valves are closed, that's going to prevent it from moving backward. So the contraction of our skeletal muscles, the contraction of even our respiratory muscles helps to kind of encourage and milk the flow of blood back to the heart. But there's really not much of a pump-like system here contributing other than these more external factors. Yes, the heart is always pumping, and yes, the blood is always flowing, but the veins need some extra support in encouraging that. When these veins become faulty or the valves become faulty, over time that can cause varicose veins. So you see here looking at the inside of a healthy vein versus a varicose. Essentially, the valves are just not doing anything, right? They're not preventing backflow of blood. So the blood will collect in these veins. They'll just enlarge over time. The walls will just kind of become deteriorated, and it creates those bumpy blue surfaces you can see sometimes on the inside of people's legs, or it can even occur in the anal canal and cause hemorrhoids as well. So some pressure has accumulated there. The valves have been destroyed, and blood is just kind of collecting within the vein. These are very commonly removed. They can cause a little bit of discomfort, or they can cause, people might feel embarrassed to have them. They don't want to wear shorts, that kind of thing. So it's very routine that these are surgically removed. And it's not a big deal because there's so many venous anastomoses or detours that the blood can take. So it doesn't even really matter if we cut these right out. So first, starting with defining blood flow. Well, it's a little hard to really give it one definition. It's the volume of blood flowing through something, right? Maybe we're talking about it in reference to a particular vessel, or maybe for an organ, or maybe for a capillary bed. It all kind of depends, right? If we want to think of blood flow in our entire vascular system, really it's equal to our cardiac output. How much blood is coming out of the heart within a minute, within its full circulation of all of the blood in our vessels. But, blood flow varies significantly depending on the organ, depending on the body's needs, right? Like I was saying, 3/4 of our capillaries are shut down at any one particular time. So blood flow really varies. In relation to blood flow is blood pressure, right? Blood pressure being the force that the blood exerts on the vessel wall, right? Any fluid that is moving has hydrostatic pressure, right? The flow of it creates pressure. Think of a hydroelectric dam, right? The idea of, collecting water, making it flow, creating pressure, collecting that flow and pressure, and creating energy. Any moving fluid has flow and has pressure. We measure that pressure in millimeters of mercury. That is just kind of the American unit that we use to measure that. And technically, it refers to the arterial blood pressure in the aorta. That's where the blood pressure is greatest, as it is surging out of the heart and into the aorta, the first large vessel. So that high pressure there is really, the top blood pressure value that we collect, and that we talk about, right? So when you go to the doctors and you get your blood pressure taken, you're having the systolic over diastolic pressure being measured. And that's a measurement coming directly from the aorta because that's the highest pressure. That's the only pressure we can really measure and collect because it is the greatest, right? 120 millimeters of mercury over 80 millimeters of mercury, the force being exerted when the heart contracts, and the force that is still present when the heart relaxes, 120 over 80, the standard, the typical measurement. That hydrostatic pressure gradient, that difference in blood pressure within the vascular system, is the driving force keeping that blood flowing. Fluids always flow from high pressure to low pressure. That's just how it is. That's physics. That's nature, right? High to low, that just makes sense. That's what keeps stuff flowing, right? If I create a lot of pressure here, it's going to move away from that. And then resistance is any opposition to flow. Fluids always meet some level of resistance, right? Blood is always meeting some level of resistance. It's going through a tube. The tube wall creates resistance, right? So there's always some amount of friction or resistance that blood is meeting. And to be more technical, we refer to it as total peripheral resistance, because most of the resistance is in the periphery, right? It's out in the systemic circulation, in the smaller vessels, and most importantly, out in the arterioles. There's not a whole lot of resistance, close to the heart in relation to far, far away from the heart, because the vessels are getting smaller at that point. So resistance is affected by three important factors: blood viscosity, the thickness of the blood, the sluggishness of it. Is it thin or is it thick? Blood vessel length and blood vessel diameter, which we're going to talk more about on the next page. So as I said, the viscosity is the thickness or the stickiness of fluid. In a healthy individual, this is going to be relatively constant. If you have a normal hematocrit, right? Normal, protein level, normal fluid, you're hydrated appropriately, the viscosity should be pretty consistent. Well, if you have an increase in viscosity, increase in thickness, it's going to create more resistance, right? It's going to be difficult for that blood to surge through the vessel if it's really thick. So we think of, polycythemia, an increase in red blood cell count, that of course will increase viscosity. And then the exact opposite, anemia, a low hematocrit, low red blood cell count will decrease blood viscosity, make it really thin. Both of those are an issue. It's not like it's better because it's thin. Anemia, there's no benefit in the blood moving quicker because it's not, spending enough time in the capillaries and allowing for adequate exchange. And then for blood vessel length, well, this too is going to be pretty constant in the adult body, right? A little different in a child as it's growing and developing, but it's a fairly constant factor, right? But anytime there's an increase in vessel length, there's going to be an increase in resistance. The longer a tube is, the more resistance it is meeting, right? There's more surface area of the wall inside, and there's more friction being produced there. And I think I'll help clarify that on the next page with an example. Blood vessel diameter is, without a doubt, the most important factor dictating blood flow and dictating blood pressure in the body, right, in any animal. This can change frequently, the blood vessel diameter, and that significantly alters the total peripheral resistance. Controlling this is the sympathetic nervous system, right, innervating the smooth muscle layer, dictating whether vasoconstriction or dilation is taking place. And, there's always an input from the sympathetic nervous system, and, it kind of establishes a normal value, or it establishes like a tone of those smooth muscle cells. But depending on a variety of factors, at times vasoconstriction can take place. So the lumen, the inside tube where the blood is flowing, is reduced because those smooth muscle cells are contracting and squeezing on that tube. That, of course, is going to increase resistance, right? And blood is going to have a more difficult time to flow through that tube, right? It's like taking a garden hose and having the water on, then picking up that tube and squeezing it with your hand or bending it, right? We constrict it, we close it off, and we may, stop the flow of water through that tube. Vasodilation, just the opposite. The vessel opens up, the lumen gets larger, more blood can flow through, right? Just like, now I'm going to release my hand from that garden hose and plenty of blood, plenty of water will flow right through. This is always happening, vasoconstriction and dilation, to move the blood where it needs to be. Very, very important. So a lot of this fluid dynamics you already understand and you don't even realize it. And we can use a really classic example here of, comparing drinking a milkshake versus drinking like a glass of water and utilizing a straw to drink those different fluids. Right? So if we think about this, okay, milkshake in the glass, which thing would be easier to drink, this milkshake or a glass of water? Well, we know that water would be easier to drink, right? Milkshake is very viscous, it's very thick, making it more difficult to drink through a straw. So that relationship is true of all fluids. There's an increase in viscosity, there's an increase in resistance, and there's a decrease in flow when it passes through a tube, right? Then we can think of vessel length or tube length, right? So ask yourself, which straw would you want to use to drink this milkshake? Well, you're going to want to use a fatter straw, which is the next property we'll talk about, but you also want to use a short straw versus a really long straw. Right? The longer the tube, the longer the straw, the more resistance that fluid is going to meet, the more difficult it is going to be to drink that milkshake, right? So the vessel length, the tube length is also an important factor. So if we decrease that tube length, we decrease the resistance and we increase the flow. And the opposite is true of that property. And then with vessel diameter or tube diameter, a fat straw is going to be much easier to drink the milkshake than a really skinny straw, right? The lumen diameter is going to be greater with this straw, much easier to drink a milkshake, and that's why we're given a large straw, right, when we're drinking a milkshake. So the tube diameter is the most important factor when we're thinking of fluid dynamics and fluid flow. So an increase in the diameter, a decrease in resistance, and an increase in flow. So you want to take all of those three, factors into consideration and think of it on this general scale, of just trying to drink something out of a cup, but then apply it to the blood vessels and blood flow. So with this image below, we're taking a look at how blood pressure changes throughout the vascular system. So on the y-axis here, we have blood pressure in millimeters of mercury, and then down on the x, it's representing the different vessels, starting with the aorta down to the vena cava. So within the aorta and in our large arteries, this is where the blood pressure is the greatest. Right? The vessels are closest to the heart here. There's going to be a lot of blood flow, meaning there's a lot of blood pressure there. As the blood flows into smaller and smaller vessels, we see that the blood pressure is diminishing. And in particular, that transition to arterioles feeding into capillary beds, we see the steepest drop in pressure. This is because the blood is meeting its greatest resistance to flow. These vessels are getting tiny, the lumen is very small, it's making it more difficult for the blood to flow through this because that resistance has increased, and that creates that pressure, that hydrostatic pressure, to really drop and reduce. Okay. This is important because we want the blood flow to slow down, we want the blood to trickle through the capillaries, allow for adequate diffusion, right, and to really allow for that exchange to take place. And then once that blood is collected in the venules to the veins of the vena cava, there's essentially no blood pressure there at all. Once that blood returns to the right atrium, we got 0 millimeters of mercury there. The pressure is completely dissipated because all that energy has been released as it's flowed through the entire vascular system. Okay, so just keep that in mind. Highest in the aorta, lowest in the vena cava, and the steepest drop being where the blood is meeting the arterioles. So here we want to focus a little closer on the arterial blood pressure. So the aorta and the major arteries are experiencing the highest blood pressure. They are the closest to the heart, so right, that makes sense. It's going to experience the greatest pressure. However, there's some variation on depending whether or not the heart is contracting or relaxing. So remember, systole, contraction, diastole, relaxation. So the systolic pressure here, the top peak here on the graph, is the highest aortic blood pressure during ventricular contraction. And that averages about 120 millimeters of mercury in healthy adults. That's kind of the statistical average. When the heart relaxes, that pressure drops to about 80 millimeters of mercury. So that's the lowest aortic blood pressure during ventricular relaxation, right? there's still pressure there. Blood is still moving forward. There's just not as much pressure because the heart is relaxing at that point in time. Okay, and then the blood pressure dissipates as it moves through the vascular system. The difference between these two values, systolic and diastolic pressure, creates the pulse pressure. Okay, this creates a pulse, a pulsatile flow of blood. So we can calculate a pulse pressure by just subtracting the systolic pressure from the diastolic, and that's the difference there. That's the moving force keeping blood moving forward, right? So when we measure blood pressure, it's really a measurement of the systolic or the diastolic pressure, and that's the true pressure being experienced in the aorta. So we can easily measure blood pressure by using a blood pressure cuff with an inflatable bulb and a stethoscope so we can hear the sounds that are produced as the blood flows. So the first thing that you have to do is wrap the cuff around the arm, and we use the brachial artery commonly to do this. So yeah, it's not the aorta, but we can still measure those high pressures. The brachial artery is not far away from the aorta either, so it's pretty easy to do that. First thing you need to do is inflate the cuff so that the pressure is greater than systolic pressure. That can be achieved by going to about 150 millimeters of mercury, 160, kind of somewhere around there. Really anything over 120 should be adequate, but we don't know if that individual maybe has high blood pressure, so you want to go a little higher than 120 to make sure that you're cutting off all blood flow through that brachial artery. So you're creating an occlusion, you're preventing blood from flowing through that particular artery. You want to make sure your stethoscope is close by, tucked right in the inside of the elbow there, and then you start to slowly release air from the blood pressure cuff. You have to do this slowly so that you can hear any changes taking place, but not so slow that, the person's arm is being starved of blood. Okay, so you slowly release the air, and what's happening is the blood begins to now flow through the brachial artery because you're alleviating that pressure in the cuff. As you're listening, you're going to start to hear kind of this knocking sound or this pulsation sound. That first sound that you hear is the systolic blood pressure. So you have to have the ability to hear the sound, but also look at the manometer and read what that pressure is at that moment in time. Okay. At that moment in time, right, the pressure in the blood has, overridden the pressure in the cuff, and we're able to measure that value, right, and say, okay, that's the systolic blood pressure. That is the greatest blood pressure within that artery. You're going to continue to hear sound as blood moves through that constricted artery, right? It's going to be turbulent and produce sound. And then it's going to come to a point where you no longer hear that blood flow. And that last sound, that last Korsakoff sound, is the diastolic pressure. Okay. Then you release the air, the blood will flow through the brachial artery, and it's going to flow very easily, so no sound is going to be heard during that point. Okay. So the first sound you hear is the systolic blood pressure. The last sound you hear is the diastolic pressure. And we can hear that and achieve that by closing off the artery, occluding it, and then hearing that blood being turbulent through the vessel. So after the systolic and diastolic pressures have been calculated, we can obtain some other values. So we can calculate a pulse pressure, which I talked about on the previous page, being a difference between the systolic and diastolic pressure. And then we can take it a step even further and calculate a mean arterial pressure, or MAP, or MAP. So this is a weighted average of the arterial blood pressure. And it's weighted, meaning that it kind of takes into account the period of time in which the heart is in systole versus diastole. Diastole lasts longer than systole, so that needs to be taken into account when calculating this MAP value. So the mean arterial pressure is roughly equal to the diastolic pressure plus the pulse pressure divided by 3. Okay. So, we're not going to dive into the math of it, but that's what it is. It's a weighted average, right? It takes into account the period of time the heart is in relaxed versus contracting. And this just creates a better representation of that arterial pressure, and it represents tissue and organ perfusion, the blood actually, flowing through the organs, okay? And if we take our typical 120/80, we calculate a blood pulse pressure, so systolic minus diastolic, that's where we're getting the 40 millimeters of mercury. We divide that by 3, add 80, we get 93 millimeters of mercury for that mean arterial pressure. Okay, this is the pressure that propels the blood to the tissues. So for the venous blood pressure, it's quite different than arterial, right? For one, the venous blood pressure is much lower, and it's steady and changes very little during the cardiac cycle. It's further away from the pump, right? Further away from the heart. And so there's not that pulsatile flow. It's fairly steady and does dissipate as you move, further to larger veins into the vena cava. So since it's a low system, there's other adaptations that have occurred to help with that venous return, to return the blood to the heart to become oxygenated again. I briefly touched on these earlier. So one being kind of the muscular pump produced by our skeletal muscles. So as we use our muscles, as we move around, the contraction action of that squeezes on the veins and pushes the blood forward, and the venous valves are going to prevent any backflow. So it's important to get up and to move around, right? That's really going to help with that venous return to circulate your blood. Individuals that are bedridden or individuals that are just always sitting down and never moving, right? The blood flow and their circulation is not as good, and that can lead to clots, especially in the veins of the legs. So it is important to move around. Also, respiratory pump, similar concept, muscles within the thoracic cavity squeezing and contracting as we breathe helps to move the blood forward in the veins. And then sympathetic venoconstriction. This just refers to the fact that, the sympathetic nervous system does innervate our vessels, right? It does cause a little bit of constriction in the vessels, and that helps to establish some pressure in that vessel and to help with return of blood to the heart. So there are 3 main factors that regulate blood pressure. We have cardiac output, total peripheral resistance, and blood volume. And for each of those factors, they have their own factors regulating them. So with cardiac output, we spent some time talking about that in the heart chapter, right? And then we were just talking about total peripheral resistance, how viscosity, blood vessel length, and blood vessel diameter really will impact that, right? So you have to look at each of these 3 factors, but also take into consideration all the other things in the background that are influencing them. So for cardiac output, right, that's the amount of blood ejected from the heart every minute. And the 2 factors that determine it are stroke volume and heart rate. Well, stroke volume is the volume ejected with each stroke, with each heartbeat. Heart rate, number of times the heart beats per minute. Collectively, that creates the cardiac output. If you have an increase in cardiac output, you're going to have an increase in blood pressure, and vice versa. Well, the stroke volume is mainly affected by venous return. How much blood is returning to the heart is going to dictate how much blood is leaving the heart, right? And then heart rate is really regulated by medullary centers. So the medulla oblongata, having cardiovascular centers that provide extrinsic control over the heart rate, how many times the heart beats. So there's so many factors here, right? So you have to have the ability to look at the big picture and think about how all these downstream effects really play a role in determining the blood pressure. Okay, we'll focus more on that neural regulation, right? I'm not going to go into detail about total peripheral resistance again because we just talked about it. So maybe you want to go, read through that again, think about that some more. The biggest factor there is going to be blood vessel diameter, regulating the blood pressure. And then, of course, blood volume. That's a simple one to understand because, the more volume you have, the more pressure you're going to have. So level of hydration plays a huge role with that. But all of these things are going to be regulated by the heart themselves, the blood vessels, and the kidneys, which is all supervised by the brain. So there's a lot of input here controlling that. We can break that regulation down into short-term and long-term. With short-term regulation, the nervous system and hormones are going to play a role in changing the blood pressure, while the kidneys are responsible for long-term regulation. So here's just a flow chart representing what I had on the previous page. So total peripheral resistance, cardiac output, being two really important factors with the mean arterial pressure. And then we expand upon that and we think of the factors which affect it in turn. So for short-term regulation, this is going to be controlled by the nervous system and by hormones that can be released during like a fight or flight response. It can alter both cardiac output and total peripheral resistance because it has the ability to affect, different body parts, the heart and the blood vessels. And for these neural controls, it's including different components. So cardiovascular centers in the medulla oblongata of the brainstem, baroreceptors, chemoreceptors, and higher brain centers can also influence your blood pressure, right? Your emotional state, if you're stressed out, if you're stimulating that sympathetic response. The cardiovascular centers in the medulla oblongata, there are, two distinct centers, first being the cardiac center, the other being the vasomotor center. So with the cardiac center, this of course is innervating the heart itself, vasomotor innervating the vessels. Okay, so the medulla can alter the heart rate, which will in turn alter the cardiac output, which will in turn affect the blood pressure. Within that cardiac center, there's a cardioacceleratory and a cardioinhibitory center. These two centers here are being controlled by different components of the autonomic nervous system. So cardioacceleratory to accelerate or increase the heart rate, that's under sympathetic control. Cardioinhibitory, that's under parasympathetic control because that effect decreases the heart rate. But these neurons, this nervous tissue is originating in the medulla oblongata, and then they travel to, the heart to either increase or decrease the heart rate. So different things are going to activate or inhibit these particular centers, which in turn affect the heart rate. Vasomotor center, this alters blood vessel diameter. That in turn is going to affect the total peripheral resistance, right? Whether the vessel is constricted or dilated, and that has a huge effect on blood pressure. And this only involves the sympathetic nervous system. Okay, so only sympathetic vasomotor fibers innervate the smooth muscle of blood vessels, with the biggest effect being the arterioles, the smaller arteries. Okay, and this vasomotor center, it always maintains a tone, so it's always providing input to, the tunica media of these vessels. And the rate in which, the signal is sent is what really dictates vasoconstriction and vasodilation. But there's always a tone, right? We have muscle tone. Our muscles are always a little bit contracted to just at least keep us upright. There's always a little contraction even when we're not even moving at all. Same concept here. If there is an increase in those signals coming from the sympathetic nervous system, that's going to activate those muscles more strongly, right? Going to cause it to contract and lead to vasoconstriction. Versus if there's weaker signals coming from those sympathetic neurons, there's less messages being sent there, less communication, that's going to relax those muscles and cause vasodilation and open up that lumen. And that, of course, in turn impacts the blood pressure. Vasoconstriction increases blood pressure, vasodilation decreases blood pressure. So for baroreceptors, these are specialized stretch receptors that we find in the aortic arch, carotid sinus, and nearly every large artery of the neck and thorax, and they're very important in regulating blood pressure. So with this image here, what we're seeing would be the baroreceptors, whether it's in the aortic arch or in the carotid sinus. And that receptor is linked to the nervous system. So there are sensory neurons there that transmit the information back to the brain. And in particular, they're sending that information to the cardiovascular centers in the medulla oblongata. So these two green lines representing sensory information going to the brain, and then red being outgoing information via the vagus nerve, so under parasympathetic control, and then blue being outgoing motor information within the sympathetic nervous system. So there's a neural reflex arc here that takes place, sensory in, medulla decides what to do, and then outgoing message to regulate that blood pressure. In particular, baroreceptors are stretch receptors. They're stimulated by the state of stretch within those vessels. So of course, if the blood pressure rises, that stretches the vessel, it stretches the receptor, and that's the sensory stimulus there, which is then transmitted to the brain. Baroreceptors are best at protecting the circulation against short-term or acute changes in blood pressure. Right? So, if you're laying down and you get up really fast, or you bend over, you're tying your shoe and then you stand up really fast, that shift in blood flow and shift in blood pressure, the baroreceptors are activated to protect that circulation and make sure that it's adequate through that transition. The location of the baroreceptor is going to dictate function and, importance. So those that are found in the carotid sinus, so higher up in the neck where the common carotid branch is, that's really going to protect the circulation to the brain, right? It's going to detect any change and influence changes to counteract that, versus those found like in the aortic arch are more responsible for maintaining adequate flow and pressure for the rest of the body. If the baroreceptor is not acting appropriately, it can cause orthostatic hypotension, so low blood pressure caused by major changes in body position. And that's just due to the failure of that baroreceptor reflex. And we're going to take a closer look at that on the next 2 pages. I'm sure everyone has felt that feeling, right? If you crouch down for a long period of time and you're doing something and then you stand right up, you feel that weird feeling like your blood is just draining right out of your body. That's just a quick temporary orthostatic hypotension. However, note what I have listed down below here. Baroreceptors are ineffective at protecting us against long-term or chronic sustained blood pressure changes. Okay, they can adapt to chronic hypertension and hypotension. So we're just talking, quick, short-term, acute changes. That's what this is providing protection against. So now we're going to take a look at how this baroreceptor reflex works when we have some increase in blood pressure. So that would be the stimulus here. More pressure, more flow, that's going to stretch those baroreceptors within the artery. That creates a stimulus, right? Those sensory receptors will become activated, and that will send that information to the medulla. When the medulla, picks up on that information, it's being told, oh, there's too much pressure, I'm really being stretched, there's too much blood in there. That's going to automatically activate cardioinhibitory centers, which is under parasympathetic control, and is also going to inhibit vasomotor centers. Okay. The effect of this is that we're going to have a decrease in heart rate, which means we're going to have a decrease in contractility, a decrease in cardiac output, decrease in blood flow, decrease in blood pressure. And it's also going to cause vasodilation because we've inhibited that vasomotor center, and that's also going to help to decrease the blood pressure. So that stimulus is automatically activating those centers in the medulla, which in turn affects, heart rate, cardiac output, and the vessel diameter, all in turn helping to reduce blood pressure and restore balance. Now we'll take a look at what happens when there's some decrease in blood pressure. That's going to mean that the baroreceptors are not being stretched, the baroreceptors are inhibited, and the medulla oblongata can sense that. there's always input being provided from these baroreceptors to the brain. So they're, taking in information stretched, not stretched, activated, inhibited, both types of information. So due to the fact that there's no stretch occurring here, that's interpreted as a drop in blood pressure. That's going to activate the cardioexcitatory center or activate the sympathetic nervous system here, and also activate the vasomotor center. So activating the cardioexcitatory center increases the heart rate, also is going to increase the contractility, more calcium is going to be released. We're going to have an increase in cardiac output and an increase in blood pressure. The vasomotor center being activated leads to vasoconstriction, and that also will contribute to an increase in blood pressure. Okay. Blood pressure will be restored by activating those two centers and to maintain that homeostasis, right? But this is a fluid thing, right? This is constantly happening and kind of finding the happy medium to establish a balance. It's not like it's on, off, right? It's just a constant kind of balance that needs to be achieved. There's always some fluctuation between these centers being activated or inhibited just to really maintain that 120/80, that typical pressure flowing through the body. So then there are also chemoreceptors, which are present, and these are found located near baroreceptors, but the stimulus here is different. Baroreceptors are activated by stretch, chemoreceptors are activated by chemicals. In particular, they can be stimulated by high levels of carbon dioxide and drops in or low pH. Remember that the blood has that very narrow range for pH, 7.35 to 7.45. If carbon dioxide levels rise, if waste products accumulate in the area, the pH will drop as well. So those are clear indicators that the blood is not circulating adequately. We need to amp some things up here and clear those waste products from the area. Okay, so if there are high CO2 levels, this is going to activate cardioexcitatory centers and the vasomotor center in the medulla oblongata. Here you can see the downstream effect. Hopefully you kind of have an idea of that by now, but ultimately going to increase the activity of the heart, constrict the vessels, and really get the blood, circulating through the vascular system more quickly to eliminate the waste, have exchange at the lungs and restore homeostasis. Now for short-term hormonal control. So we were just talking about neural control, now thinking about how hormones, chemical messengers can influence the blood pressure kind of temporarily or for the short term. Epinephrine and norepinephrine from the adrenal medulla are released during a sympathetic or a fight or flight state, right? So when we're really scared, we're really nervous, that kind of adrenaline rush that we feel is due to the release of epinephrine and norepinephrine. The effect on the heart is that it increases the heart rate, increases the contractility by making more calcium available to the cardiac muscle cells, and that in turn increases cardiac output and blood pressure. Okay. Angiotensin 2, ADH, and aldosterone, I'm going to talk about that on the next page, but those three independently have an effect that increases blood pressure. And then for the opposite, the antagonist here is atrial natriuretic peptide, or A&P. This is produced by the atria, right? It's right in the name, atrial, and it's produced in response to high blood pressure. So if there's high blood pressure, that's going to be felt, by the heart. That's going to increase the afterload, increase the back pressure on the heart, and that stimulates the atria, and they release this atrial natriuretic peptide to help to counteract that. So it functions to decrease blood pressure. It antagonizes aldosterone, the sodium-retaining hormone, and it causes widespread vasodilation to help to quickly, reduce blood pressure. But notice how that's different than the rest of these here. The ones listed above function to increase. Atrial natriuretic peptide decreases blood pressure. Then for long-term regulation, this is dependent on the kidneys. The kidneys are responsible for filtering our blood, around 150 to 180 liters of blood a day. That's just amazing, right? Always filtering the blood, so it's very intimate with it, and it can always, sense, detect any changes in blood pressure, and is responsible for regulating that in the long term. Okay. One way that the kidneys will regulate that is in a direct manner by, really influencing urine output. And then indirect renal mechanisms is involving our hormone cascade of renin, angiotensin, aldosterone. Okay. So for the direct mechanism, we can directly alter blood volume by the amount of urine that is produced, right? So you drink a bunch of water, well, that's going to lead to an increase in blood volume because you just drank a bunch of water, osmosis and all that good stuff. More fluid is going to accumulate in the blood. And, the kidneys will detect that. Okay, we have an increase in blood volume. I'm going to start to make a bunch of pee, right? A bunch of urine, and help to reduce that blood volume, and vice versa. So urine output directly influencing blood volume, directly influencing blood pressure, right? With the indirect renal mechanism, hormones are playing a role in doing basically the same thing, but it's more of an indirect manner because it's dependent on, these hormones. But ultimately functioning to, retain certain substances and retain fluid to help maintain an adequate blood volume and blood pressure. I'm going to use the diagram on the next page to explain how that all works. So here we're taking a look at the renin-angiotensin system. Very important for when there's any drop in blood pressure, drop in fluid volume. And as you should see here, it involves several organs, and it's a very complex, thing that takes place. All starts with the kidneys. The kidneys can sense when there's a drop in blood pressure and a drop in blood volume, and that causes a release of renin from the kidneys. Renin would be released into the bloodstream and act on angiotensinogen, a precursor protein here, produced by the liver, always just kind of circulating around in the blood. Renin converts that into angiotensin 1. Angiotensin 1 floating around the bloodstream will interact with ACE enzymes, angiotensin converting enzyme. That enzyme is produced by the lungs. That converts it into angiotensin 2. Angiotensin 2 is an extremely potent substance. Okay. It will directly cause vasoconstriction. So see at the top here, very potent in doing that. Immediately causes vasoconstriction, narrowing to help to increase the blood pressure. But what it also does is acts on the adrenal gland and causes a release of aldosterone. So aldosterone, that is the salt retaining hormone. It causes a retention of sodium by the kidneys. In turn, that will cause a retention of water, right? But ADH is also going to be released along with this, and that in too is going to cause a retention of water as well. That retention there of sodium and water is going to increase the blood volume and help to restore or, maintain any blood pressure that it can, right? So the effect collectively here is all to increase blood pressure. But see how it involves multiple chemical reactions for this to take place. And when something is multi-step like this, that's very important because that means we have a lot of regulation. We don't want this happening at inappropriate times. So, all of these steps are there for a reason to make sure it only happens when absolutely needed. So then here's the big picture, right? There's many different things that can occur, many different factors that all affect the mean arterial pressure, right? So at the top here, we have different stimuli that can influence that, and kind of the downstream effect or the physiological response that will occur and the result that will take place. So, it can be hard to really take all these different things into consideration, but the reality is that all of these things really influence our blood pressure. So you want to spend some time with this, make sure you understand all these different scenarios and how it plays a role in affecting the blood pressure in the body. Hypertension is the term we use to describe an individual that has chronic high blood pressure. When I say chronic, I mean that, every time that individual's blood pressure is taken, it's over 140 over 90, right? Every time they go to the doctor's office or whatnot. Not just an acute scenario where you're really emotionally upset or something and your blood pressure spikes. So it's ongoing, reoccurring. It's estimated around 30% of people over 50 are hypertensive. And, going right along with that, there's going to be cardiovascular disease. So hypertension is a silent killer. It's fairly asymptomatic. You may not know you have it. It can be ongoing for a while before you have other downstream problems. But the reality is that it's making the heart work harder. There's more pressure. There's more resistance. So it really disrupts the pump, right? It makes everything malfunction. Okay, so prolonged hypertension is going to be the major cause of heart failure, vascular disease, or renal failure, right? Because now you have more blood passing through the kidneys. So that's an overload for those delicate organs and can increase your risk of stroke and heart attack. So something that really needs to be checked yearly, make sure you don't have it. And if you do, you want to be taking medication to counteract that because you don't want to overwork your heart and other vital organs. It can be classified as either primary or secondary. With the primary or essential hypertension, this is most of the cases. So there's not like one particular thing causing it. It's a variety of things, lifestyle, the kind of environment you're in, what kind of foods you eat. Are you overweight? Are you elderly? Do you have other diseases? Are you a smoker, right? So several things kind of playing a role in this. So one major way to counteract it is just to change your lifestyle, right? Reduce your sodium intake. That's really going to reduce your water retention, your blood volume, and your blood pressure. Restrict fats and cholesterols. Start exercising, lose some weight. So you can turn this around by just having a drastic change in lifestyle. Medications are going to be used as well. Beta blockers, which actually will reduce heart activity a little bit. So it reduces the contractile strength to help counteract pressure. Calcium channel blockers, same kind of thing, kind of reducing the contraction occurring within the muscle cells. And then there's ACE inhibitors as well, playing a role in the renin-angiotensin mechanism. With secondary hypertension, this is because there's some identifiable condition or cause. Maybe something is obstructing the renal, the kidneys. Any kind of kidney disease is definitely going to cause hypertension because the kidneys are responsible for, monitoring the volume of blood, right? And if there's too much, that's going to tell the kidneys to excrete that water. So if that whole system is messed up, then it will definitely lead to hypertension. Atherosclerosis is another cause of hypertension and it also increases the risk of heart attack, stroke, and vascular disease as a whole, embolisms and things of that nature. And this is when there's a fatty buildup in the arterial walls. So different triglycerides, cholesterols will embed within the wall and lead to these hardened plaques. So this is going to restrict blood flow in the immediate area. It's going to increase the hypertension in that area as well. So not only cutting off blood flow to the organs and tissues downstream, but it's also increasing the pressure on the backside, right? It's like taking a hose full of water and squeezing it and kinking it, right? It really just disrupts the flow. Not good for the body, right? Not good for the engine here. And this is quite a common thing in our population, especially in the United States, right? So this is, one of the major things when we think of cardiovascular disease. Hypotension is low blood pressure, persistently below 90/60. So again, this is more like a chronic scenario where every time your blood pressure is taken, it's on the lower side. Well, typically really not an issue. You might just be statistically lower than everyone else, and, just a variation, not really a huge concern there. However, when there's an acute situation, meaning a sharp drop, an immediate drop in blood pressure, that's a dangerous scenario, right? There's a drastic fluctuation at that point. So it could lead to an inadequate flow of blood to certain tissues, making them hypoxic, right? Starved of oxygen. The tissues could die, become necrotic. So that's not good. And this would be an important sign of circulatory shock. Maybe someone is bleeding internally, right? Blood is pooling and filling up their abdominal cavity. They were just in a car accident or something like that. So an immediate drop in blood pressure would be an indication of that. And then, that individual is at risk of going into circulatory shock because there's not enough blood in the system, right? In the engine to work. Chronic hypotension can be associated with other diseases and disorders. I'm not going to spend a whole lot of time on that. But one I do want you to consider is orthostatic hypotension. So this is when there's a quick drop in blood pressure due to a change in body position. So this happens to me, I guess I could say kind of frequently. For instance, I'm working in the garden, I'm pulling weeds, or maybe I'm mulching a bed and I'm crouched down, right? My knees are bent and all the blood kind of starts to pool at the lower part of my body. If I stand up really quick, I feel that, kind of dizzy, weird feeling. And that's because I've had an orthostatic hypotension kind of episode where there's a sudden drop in blood pressure because that blood is quickly moving to a different area. The dizziness is associated with a little bit of a drop in oxygen delivery to the brain. And this is common more in elderly individuals when their baroreceptors stop to function and be as reflexive as they used to be. But, this could happen to anyone. And really, it's just because of your body position influencing this. Circulatory shock is any condition in which the blood vessels are inadequately filled. There's not enough fuel in the engine. The blood cannot circulate normally. The blood flow is not meeting the tissue needs, not meeting organ needs. The body goes into shock, right? There's a couple different types: hypovolemic, vascular, or cardiogenic. So just as the name implies, hypovolemic, there's not enough volume, right? This is going to be the most common scenario. And I was referencing this earlier. So say someone gets in a car accident, right, and they're bleeding internally, or there's a gunshot wound and there's some large-scale blood or fluid loss. This is going to lead to hypovolemic shock. Vascular shock, this is when the volume is normal, but the circulation is poor due to extreme vasodilation. So some area in the body, the vessel has dilated way too much. Blood is collecting in one particular area, and it's just not circulating through all the tissues like it should be. So this could be anaphylactic. So this could be due to a severe allergic reaction. Could be neurogenic, failure of the autonomic nervous system to maintain that vasomotor tone of the vessels. Could be septic shock, a severe bacterial infection that's spread to the bloodstream. Typically, the type of bacteria that causes septicemia or septic shock are notorious vasodilators. So they release toxins that cause that dilation. Cardiogenic, cardio heart, right? This is pump failure. So now the pump is no longer circulating the blood like it should be. The heart is shutting down. Circulation is not sustaining the body needs. The term tissue perfusion refers to blood flow through a particular tissue or through a particular organ. And it is always changing, okay? It's dependent upon your body's state and what you're doing. Are you sitting on the couch and relaxing and your autonomic nervous system is dominant at that time? Or, are you on the treadmill running and your sympathetic nervous system kind of kicks in? So your blood is going to be moved to the area that needs it the most, right? And the rate of blood flow to each of those tissues and organs is nearly the perfect amount for proper function. No more, no less. So blood is not wasted on cells and tissues that don't really need it. There's different controls or different mechanisms that regulate blood flow and regulate perfusion. We have intrinsic controls and we have extrinsic. We'll talk more about that. But the main idea here, intrinsic, is within. So there's some auto-regulation with that, local controls in that immediate area, and has all to do with changes in the diameter of arterioles, right? Influencing that flow to a particular location. Versus extrinsic, meaning outside, right? So the sympathetic nervous system and hormones, overall body systems influencing that regulation. Here's a visual for you thinking about the distribution of blood flow at rest versus strenuous exercise. So here we're thinking of total blood flow to different organs at rest, sitting on the couch, relaxing at the end of the night. We have quite a bit of blood flow going to the abdomen, our GI tract, or, digesting food maybe, absorbing nutrients. The kidneys are receiving a lot of blood, filtering our blood for us, producing urine. The brain is going to be receiving a constant rate no matter what. And then with the skeletal muscles, a decent amount there. But again, you're really not doing a lot of physical activity at rest. Then say, you're like, all right, I got to go outside and play some basketball. I've been lazy all day long. Well, once you start increasing your activity there's a drastic change in tissue perfusion. Blood is going to be shunted to different parts of the body because you're doing something different, right? So we can see a drastic increase here in blood flow to the skeletal muscles. And the skeletal system is capable of receiving over 70% of the blood volume at any given time, right? Really needing to supply those muscles to cause contraction. As I said, to the brain is going to be consistent no matter what. There's going to be an increase going to the heart, decrease in, our kidneys and abdomen, because at that particular time, your body's not worried about producing urine or digesting food, right? You're moving about, your muscles need the oxygen and ATP. I want to talk a little more about intrinsic control of blood flow. So this is that auto-regulation within a cell, within that immediate area, okay? It's independent of, the nervous system, independent of hormones, right? It's acting on its own. And overall, the only thing that's really changing is the diameter of local arterioles supplying capillary beds in that immediate area. There's metabolic controls and myogenic controls in terms of short-term regulation. And with the metabolic, this is when the cells in the area start to, sense that there's a change in oxygen levels, there is an increase in metabolic waste accumulating in the immediate area, and it's basically a red flag, like, I'm kind of being starved of oxygen here, and all of this waste is building up. Something needs to change. And what happens is there's going to be direct relaxation of the vascular smooth muscle to dilate the blood vessel in that area, open it up a bit, and allow more blood to come to the area, flush away the waste, and deliver more oxygen. Nitric oxide is released from those arterioles to cause that vasodilation, and that helps to increase that tissue perfusion, increase that blood flow to the area. So again, this is an auto-regulation, kind of an automatic thing happening within the tissue itself. Then for myogenic, I always think muscle. So smooth muscle cells have this ability to automatically respond to changes in blood pressure. So if a smooth muscle is stretched, it automatically contracts. So say within the immediate area, within a tissue, the blood pressure rises sharply. There's a high blood pressure. Well, that's going to stretch smooth muscle cells in that arterial, in that capillary bed, and it causes automatic vasoconstriction. The smooth muscle cells sense that stretch and go, I'm going to constrict. I'm going to decrease the diameter here and decrease perfusion. So this makes sure that the engine is not over-flooded, that those cells are not over-flooded with blood. And just the opposite is true as well. So if the cells and the tissues in the immediate area start to sense there's too much of a drop in blood pressure, they'll actually vasodilate. They don't have that stretch stimuli. They'll relax, open up to try to increase perfusion. So with this myogenic control, it's all about maintaining balance. And if there is an imbalance of too high or too low, these smooth muscle cells will counteract that to maintain homeostasis in the immediate area. And then thinking about more of a long-term autoregulation, something that can occur within a tissue is angiogenesis. So this would be the synthesis of new blood vessels in the immediate area. So if a tissue is constantly, being starved of oxygen and nutrients, well, that tissue is like, well, I got to do something about this, right? And it'll start to synthesize new blood vessels, more delivery of blood in a more of a long-term manner. However, one downside to this mechanism is that cancerous cells utilize this as an advantage to become larger. So once a tumor actually begins to form, if new blood vessels are being synthesized within that tumor, not a good scenario at all. So they've kind of hijacked this interesting mechanism. We'll talk a little bit about blood flow in special areas. I already kind of talked about the skeletal muscle. I think you understand that depending upon activity, there's going to be an increase or decrease of flow to our skeletal muscle. With skin, this plays a role in regulating our body temperature. So our cutaneous circulation, blood vessels out in our skin, depending upon if we're hot or cold, blood will either be shunted to or away from the cutaneous circulation to help control our overall body temperature. For the brain, there's a constant level of blood flow to it. The brain cannot tolerate any level of ischemia, inadequate blood supply or blood flow to the brain. The brain cannot store nutrients, and it is highly metabolic. It uses a lot of energy, a lot of ATP, a lot of glucose, so it cannot tolerate any change in perfusion. Syncope refers to fainting, and this can occur if the arterial pressure falls below 60 millimeters of mercury. So if there is a drop in pressure and blood flow to the brain and there's any small amount of ischemia, you'll faint and pass out. And the concept there is that your body's trying to get into a flat position, like a laying down position, so it's easier for the blood to get up to the brain. If you're standing up, it requires more effort for that blood to get up to the brain. The opposite of that could be an increase in the mean arterial pressure above 160 millimeters of mercury. This will cause swelling of the brain or cerebral edema. Right? There's too much pressure. There's too much fluid being pushed out on the arterial end of a capillary bed, and fluid will start to collect in that area. The brain doesn't have a lot of room for extra fluid, so that's not a good scenario either. And then stroke or cerebral vascular accident, a CVA, kind of a fancier term for stroke. That's when there's sudden brain tissue death due to ischemia, because a particular area of the brain did not get enough oxygen, enough nutrients, and it immediately starts to die. So a common cause of this is atherosclerosis, fatty plaque buildup in the arteries supplying the brain. Could be thrombosis, so an internal blood clot, or an aneurysm, bulging of the blood vessel wall that creates kind of an out pocket that could burst open and cause someone to bleed out in their brain. For the lungs are kind of interesting in terms of tissue perfusion. Their autoregulatory mechanism is actually opposite of what we find throughout the rest of the body. And you have to keep in mind that the function here is the opposite as well, because the lungs' function is to oxygenate the blood. So actually, tissue areas where there's low oxygen levels, vasoconstriction occurs in the immediate area. So it closes off that area. It doesn't want blood to go to that immediate site because there's not a lot of oxygen there for it to pick up. Versus high oxygen areas, high oxygen levels is going to cause vasodilation, going to cause blood to go to that high oxygenation area to pick up as much oxygen as it can. Okay, so this really allows the blood flow to oxygen-rich areas for the lungs to pick up as much oxygen as it can and deliver that to the rest of the body. Then with the heart, so what we're finding here in terms of special blood flow, well, of course, we have that separate coronary circulation, right? And during strenuous exercise, this blood flow to the heart muscle can increase about 3 to 4 times. It's always having a pretty consistent flow to it, but we do see that change depending upon physical activity. The flow of blood through the body is the slowest at capillary beds. And when we look at this image down below, when we look at these two graphs, on the bottom here we have velocity of blood flow. So it's the highest in the aorta, stays fairly high in the major arteries. Then there's a steep drop in velocity, and it's the slowest in those capillaries and capillary beds. Well, the reason for that is because there's a lot of surface area. When we think about an arteriole feeding into a capillary bed, look at all of these different tiny vessels in which the blood has to travel through. So the total area, surface area that blood needs to be distributed throughout is the highest in the capillaries, so the flow is the slowest. And this is really important. This is really beneficial because it allows for adequate time for exchange between the blood and the tissue cells and the tissue fluid. If blood was just flying through the capillary bed, nothing would benefit from it, right? There would not be enough time for diffusion and osmosis to take place. So the blood flow through, it can also be described as intermittent. So, up and down fluctuations due to vasomotion, due to on and off vasoconstriction and vasodilation of the arterioles, auto-regulating the tissue and cells needs. Right, so this ties back into that whole tissue perfusion concept we were just thinking about, right? What's the body's state? What is it doing? Where does the blood need to go? So there's on and off regulation that will occur and really influences where the blood is going, which capillary beds of the body. Capillary exchange refers to the movement of solutes between the capillary and interstitial fluid, so across the capillary wall. We look at this image down below here. So this would be the lumen, the inside of the capillary. Here's the capillary wall, endothelial cells forming that, and then we have a thin basement membrane. And then the blue would represent interstitial fluid, tissue fluid between the tissue cells. So the exchange is across the capillary wall. And there's different types of transport that can take place. We could have simple diffusion directly across the endothelial cell. So oxygen and carbon dioxide move in this manner. Movement between the cells due to intercellular clefts, so like glucose and amino acids need to move in this way. Some capillaries are going to have fenestrations, especially like in the kidneys, just pores essentially that allow for solutes to move. And then our larger molecules that can't squeeze between the cell or through the cell are going to depend on vesicular transport. So a vesicle needs to be formed to move across the capillary wall. Bulk fluid flow refers to the flow of fluid that moves about during capillary exchange as blood is flowing through the capillary beds all throughout our body. So on the arterial end of a capillary bed, the fluid is flowing out. That's due to a lot of hydrostatic pressure. The blood pressure pushes the fluid out, pushes solutes out on the arterial end. On the venous end, we have reabsorption, recollection of, a lot of that fluid. That's due to osmotic pressure, due to the components within the capillary wall at that point, that sucking force pulling the water back in on that venous end. So if we consider the entire body, we consider all the capillary beds throughout, we have about 20 liters that flow out on the arterial end, about 17 liters being reabsorbed on the venous end. So we have 3 liters that's kind of lost in the interstitial fluid. Fortunately, we have the lymphatic system and the lymphatic capillaries that reabsorb that lost fluid. The fluid then becomes what we call lymph. It passes through all the lymphatic vessels, through lymph nodes. The fluid's monitored and checked out by our immune system, and then that fluid returns to the general blood circulation. So on a bulk fluid flow, kind of referring to a large scale fluid, right? The different compartments that it's in the body. And that's very important for determining blood volume as a whole, blood pressure as a whole, and can influence edema or tissue swelling in an immediate area due to any changes in this bulk fluid flow and capillary exchange. So hydrostatic pressure and colloid osmotic pressure are the two main factors that really influence bulk fluid flow and really influence net filtration pressure at a capillary. Hydrostatic pressure is the pushing force. Any fluid that is moving has hydrostatic pressure, right? Think of a hydroelectric dam. It functions because water is moving and passing through it. It turns a turbine and it makes energy, right? It has hydrostatic pressure. On the arterial end of a capillary bed, there's absolutely hydrostatic pressure. Blood is flowing in and flowing through because the heart is pumping it and moving it forward. So this is the force that's really influencing the outflow of fluid, the outflow of solutes on the arterial end of a capillary bed. It literally causes filtration through the capillary wall. Versus colloid osmotic pressure, this is kind of almost the opposite. This is the sucking force, kind of the pulling in, the reabsorption that takes place on the venous end of the capillary wall. And this works because there's still stuff trapped within the capillary on the venous end. Not everything can pass through the capillary wall, right? There's still going to be plasma proteins inside, red blood cells, white blood cells, large molecules trapped within that capillary. And those have attractive forces, right? Water moves towards stuff. So there's high osmotic pressure on the venous end that sucks the water back in. But as we saw on the previous page, not all of it, right? But does reabsorb most of the water. When we use the term net filtration pressure, we're just talking about the difference between hydrostatic pressure and osmotic pressure. What are the net forces acting at a capillary bed, right? What's the out, what's the in, what's the difference there? That really is what determines where the fluid is and where it should be. So here's a better visual of that. We have arterial shown on the left, venule on the right, capillary right in the middle. So when blood is flowing through this arteriole, there's a decent amount of hydrostatic pressure there, right? That's the pushing force on this end of the capillary. It's going to push fluid and solutes out of the capillary to supply those tissue cells. Well, keep in mind, within that capillary, we still have red blood cells, plasma proteins, other solutes trapped within. When it reaches the venule side, there's a lot of osmotic pressure. There's that sucking force that draws the water back in on this end. So not only do we have diffusion, we have filtration due to that hydrostatic pressure and then reabsorption on this end. But do realize not all of the fluid is reabsorbed, right? Look at our net filtration pressures, 10 out, 8 in. So some fluid is lost to the interstitial space, but we do have those lymphatic capillaries there to reabsorb that lost fluid. So edema can occur when there's any alteration in net filtration pressure at a capillary bed. There's different causes for this. One could be an increase in capillary hydrostatic pressure. So we have an increase in the blood pressure here, increase in that pushing force on the arterial end of a capillary, right? So there's a lot more than just 10 pushing out, maybe there's 20, right? That's going to cause too much fluid to hang out in that tissue, and the body's not going to be able to keep up with the extra fluid that it's accumulating. Or what we could have is an increase in interstitial fluid osmotic pressure. Maybe, the environment out in the tissues is becoming hypertonic and it's drawing water out of the capillary, right? We're going to have an increase in interstitial fluid there. There's going to be tissue swelling. We call that edema. Also could be a scenario in which there's a decrease in capillary colloid osmotic pressure. Maybe there's not enough stuff in the blood, not enough plasma proteins in there, not enough sucking force acting at that venous end and not drawing the water back in toward the venule. So different scenarios, but it's all the exact same idea. And you got to think back to osmosis and different solution osmolarities and how the water moves about. Water moves towards stuff, right? It likes that high solute concentration, and that will really influence the bulk fluid flow, really influence the exchange that takes place at our capillaries. And then what I put at the bottom here, which would be kind of different than the topic we're discussing right now, but edema will also result if there's any blockage of the lymphatic system, right? This can occur due to cancer, a tumor may be forming within the lymphatics, but this is just where, there's a buildup in the draining system, right? The lymph can't collect that lost fluid, so it starts to build up at a tissue site. So we've reached the end of chapter 19, the blood vessel chapter. We're going to focus on identifying and labeling the vessels in lab. So more of the visual component is going to come into play there. And we'll also be talking a little more about like blood pressure and things like that in relation to our vessels.